So the area of science with genomics is just really exploding. And now that we know the human genome, we're able to study other things. And we're going to call them omics. Anything, areas of biological research that have this omics connection are continually developing, and we're just studying things like proteomics. This is going to be the studies of all the proteins of an organism. Metabolomics, metabolic problems and issues. Metagenomics, transcriptomics, all these sorts of things are going to be areas of huge study. So this big data, this is, if you are good with computers and you know the science behind it, there are a lot of jobs out there for you. Okay, so this area, the era of big data is here. So Nutrigenomics, new field, completely new field, focuses on understanding the interactions between your diet and your genes. They're going back looking at Stone Age genomics, using small amounts of ancient DNA from bone and other tissue. They're using these to study evolutionary relatedness of various extinct and present-day species. We're going to talk in a second about T-Rex. The Personal Genome Project. We talked before about the price of sequencing an individual human genome. Still is about $1,000 to about $4,000. It's dropping though, and it's not unforeseen that in the next 10-20 years that's going to drop to about $100 a person. As technology advances, it becomes cheaper and cheaper and more available to the masses. The Human Microbiome Project is a really cool project where they were looking to see all of the different microorganisms that are associated with the human in all of the different tissues. So within the intestines, within the skin, in the mouth, everywhere on the human body. There are more cells of bacteria on us than there are human cells. And there are more genes being expressed by these microorganisms than there are human genes, by a factor of about 10 to 100-fold. So this is not insignificant, and they can definitely participate in the health of an individual. There have been studies showing things like mice who have a particular type of microorganism in their gut remain thin. When other mice have a particular type of microorganism in their gut, they tend to remain obese. So things like this may be playing a role with the increased prevalence of obesity in America. Comparative genomics is where you're going to compare the genomes of different organisms in order to answer questions about genetics and other aspects of biology. When we look and we compare humans to other species, the percentage of genes that we share is pretty high. So it ranges from about 30% of the genes in yeast. Remember, yeast are these single-celled little eukaryotic organisms, yet our genes are 30% the same, to about 80% similar in mice, another mammal, and about 98% similar in these chimpanzees. So again, we are very, very closely related. A lot of people don't like to admit how closely related we are to our nearest relatives. Many of these mutated genes involved in human diseases are also present in these model organisms. And so we can study these in these other model organisms. Going back to the comparative genomics, still this rough draft of the Neanderthal genome shows that about 2/3 of the genome were similar. Comparative genomic analysis will help identify areas in the genome where humans have gone rapid evolution since diverging from these Neanderthals. And so these studies suggest that this interbreeding took place maybe about 45,000 to 80,000 years ago. And the genome of these non-African Homo sapiens contains about 1 to 4% of the sequence inherited from these Neanderthals. So there definitely was some interbreeding going on between humans and Neanderthals way back when. Metagenomics, or environmental genomics, is the use of this whole genome shotgun approach to sequence genomes from entire communities of microbes. And they can look in samples of water, air, soil. Pretty cool. The general method, you just take up some soil or you take some water, whatever given environment, and you just sequence to see what's there. You try to then apply it back to what microorganisms do you think were there. The thing is, a lot of these microorganisms are very difficult to culture. So they may be present in the soil, they may be present in the water, but it's very difficult to study them because we can't culture them. We can't grow up a culture, they can't grow on a petri plate, they just only can grow in the water or only can grow in the soil. When you try to bring them into the lab, they die. So we still, though, can study them if we can use this sort of technique where we can just sequence them where they're at and get the information from that. This will tell us about the millions of species of bacteria as well as viruses, particularly bacteriophages, which are viruses that infect bacteria. Why do we care about this? Well, this is our best way to identify genes with novel functions. Things like potentially new antibiotics or other drugs, may have valuable applications in medicine or biotechnology. We don't know what we don't know until we can sequence and see what's out there. Transcriptome analysis, or global analysis of gene expression studies, look at the expression of genes by a genome. So you can qualitatively identify which genes are there, which ones are not, which ones are being turned on, which ones are not. And then you can quantitatively measure it by looking at the levels of these expression. So not only can you say, yes, it's on, no, it's not on, you can say, well, at what level is it on? Is it, on really high level gene expression? Is it dropping very low? Is it somewhere in between? Microarray analysis is a great way to do this. So what it does is it enables researchers to analyze all of a sample's expressed genes at the same time. So these microarrays, also known as gene chips, they're just these little glass slides, and they have DNA molecules attached. A single microarray can have over 40,000 different spots of DNA. The last time I looked, the human chip was about 47,000 spots, I think, on that chip. We know that there's only 20,000 genes, or about, give or take. So a lot of them are on there more than once, and so that provides us some redundancy when you're looking at whether or not a particular gene really is being turned on or not. So the way that it works, you have a tissue sample or a cell sample, and usually you're going to be comparing two samples. Maybe control samples to samples that were treated with a drug or treated in some sort of a way. You isolate the mRNA from that. You make cDNA through this reverse transcription like we talked previously. You then apply this mixture to a DNA microarray. These are the little spots that would be attached to this chip. If you were to zoom in and look at one of those particular spots, what you could see is this basically acts like a little bait. And then hopefully, if your cDNA is present, it will bind to there. If it's not present, it can't bind there. You rinse off the excess, you put it into a machine, the scanner will read the spot to see where are things binding, where are things not binding. If you see no fluorescence, that means your gene is not expressed in the tissue sample. Moderate fluorescence, maybe low gene expression or medium gene expression. Bright fluorescence, it's going to be very highly expressed gene in your sample tissue. So when you're doing these, you really need to compare it to something, right? You need your control cells or control tissue samples that you're comparing it to. What you can then see is the level of those genes. Is the level of the gene increase upon treatment? Does the level of that mRNA decrease upon treatment, or does it stay the same? This can then tell you genes that are either being downregulated or upregulated given various different conditions. We can also go back and we can figure out what are those genes involved with. Are they involved in the cell cycle? Are they involved with this particular pathway or that particular pathway? And you can really follow to see what's going on. A lot of really powerful information out there from these. Systems biology is going to incorporate the data from all of these different areas, from genomics, from these microarray analysis, proteomics, other areas of biology. And the goal behind it is really to try to figure out how is everything connected. Everything's connected, everything's related to one another, all of these pathways really come together. And so trying to do things like making a network map, it's basically just a sketch and it shows these different interacting proteins, genes, and other molecules. So you can see here, this is one for humans. It's definitely not complete. And the different colors are things that are involved with different tissues or different areas. So like this blue is a bone, this light green here is ear, nose, throat, this light blue is endocrine, this red is metabolic, and so on. Okay, and you can see different diseases on here. Deafness, very highly related to the ear, nose, and throat for obvious reasons. However, there are other areas which are related which you might not have realized. Dermatological. So dermatology, how is that related? Well, there's different genes involved with the skin that may be involved with this sort of function.